Dear viewers, welcome to my channel Enzymes. I am Dr. K. Sheila, Assistant Professor in Biochemistry, Sacred Heart College, Autonomous, Tirupattur. Enzymes may be defined as biocatalysts synthesized by living cells. They are protein in nature. It's a thermolayable in character and specific in their action. The biocatalyst of enzymes by living cells, they increase the speed of reaction. The word enzymes mean in East, the branch of science dealing with enzymes is called enzymology. In 1878, Curie used the word enzyme. All the enzymes are protein, but all proteins are not enzymes. Introduction. Enzymes are biocatalysts that speed up the rate of the biochemical reaction. Most enzymes are the three-dimensional globular protein, that is a tertiary quaternary structure. Some special RNA species also act as enzymes and are called ribozymes. Structure of enzyme. The active site of an enzyme is the region that binds substrate, cofactors and prosthetic groups and contains residue that helps to hold the substrate. Active sites generally occupy less than 5% of the total surface area of an enzyme. Active site has a specific shape due to tertiary structure of protein. A change in the shape of protein affects the shape of active site and functions of the enzyme. Active site, always they having the two type of the site. One is a binding site and catalytic sites. The binding site, it chooses the substrate and binds it to active site. Catalytic site, it performs the catalytic action of an enzyme. Cofactors. Cofactor is the non-protein molecules which carries out of chemical reaction that cannot be performed by standard 20 amino acids. Always the cofactors, they are the two types. One is the organic cofactors and inorganic cofactors. So what is the role of cofactors? It's a conversion of inactive protein to active protein. So always the enzyme having the binding site as well as a catalytic site. The binding site, they are helpful for the bind to the cofactor into the inactive protein. And after the binding, they are involved to the catalytic process. They are conversions of inactive protein to active protein. What is the nomenclature of enzyme? When enzymes were first discovered, they were given various unsystematic names by their discoveries such as pepsin, trypsin, thylline, zymase. So like a pepsin, trypsin, it is a proteolytic enzyme. So the substance on which enzyme it acts. Based upon the substance based, the enzymes will name like a esterase, which acts as a ester of the substrate molecule. Amylase, which act as a starch. Urease, which act as a urea. The based upon the substrate, the name it's called as the naming its form enzyme. Furthermore, the name of the some enzyme also denotes the type of the reaction. The based upon the reaction, the name it's called as a enzyme's name, like a lactic acid dehydrogenase. This enzyme catalyzes the dehydrogenation. The dehydrogenation DE, that is indicates the removal of hydrogen of lactic acid. Some enzymes were named before these general rules were formulated. Such enzymes have retained their tribal names like pepsin, trypsin and all the things. So there is no perfect method for the classification of enzyme. In 1961, by the Enzyme Commission of the International Union of Biochemistry to introduce some of the classifications of enzymes. Next we'll move on to lock and key mechanism. The lock and key mechanism is nothing but it is reactions between the lock and key mechanism. A lock and key analogy may be used to describe the fundamental action of single substrate enzyme. In this case, the enzyme is the lock and the substrate is the key. Enzyme is the lock and substrate is the key. Only the correct size of the key, which is the substrate, enters the key hole, which is the active site of the lock, which is the enzyme. The enzyme is always the lock. The substrate is the key. So by the use of the key, they open to the lock. The enzyme having the two sides, one is the binding site as well as the catalytic site. This binding and catalytic site, they are opened by the use of the key. Other keys that are too tiny, too big, or wrongly positioned do not fit the lock. Only the right shaped key can open the lock. This diagram shows that the key it acts as a substrate, lock, enzyme. They may be its correct size and shape means the lock and key it will be fixed, and the complex is called enzyme substrate complex, lock and key complex. This diagram shows that it's a correct key and lock will be present. So they are easily binds. They are finally productions of enzyme substrate complex, and also they called lock and key complex. Induced fit theory. It is one more another types of the mechanism process, but it's small modulations from the lock and key mechanism. Koshland modified the Fisher lock and key mechanism in 1963 in the form of induced fit mechanism. The essential features of this model is the flexibility of the region of the active site. The active site always they present in the enzyme site. Unlike the lock and key mechanism, according to this theory, the active site has a shape complementary to that of the substrate only after the substrate is bound. Like that enzyme does not retain its original shape and structure complementary to that of the substrate, but the contact of the substrate induces some conformational or geometrical changes in the active site of the enzyme molecule. Consequently, the enzyme molecule is made to fit completely the configuration and active center of the substrate. This process is dynamic recognition, so it's called as a induced fit. The substrate and as well as the enzyme it is a log. The log it does not consist of any specific shape, and also the substrate it's also not correct shape of the key. So first it will change some structural or some chemical reactions it's occur. After the forming of the correct shape and key, they are easily fixed to substrate and enzyme. They finally, once they are fit in the correct shape, they are fitting of enzyme as well as a substrate. It's called as a enzyme substrate complex. Next, we'll move on to factors affecting enzyme activity. The sum of the factors to affect the enzyme activity, like a temperature, pH, substrate concentration, all the things. The rate of chemical reaction or increase as the temperature is raised. Rise in temperature causes increase in reaction rate in enzyme catalyst reaction. Certain temperature commonly known as a optimum temperature. It's mostly usually in the ranges 37 degrees Celsius to 45 degrees Celsius. Above which enzyme is being proteinous nature in undergo denaturation with loss of activity. It's due to loss of secondary and tertiary structure of the protein moiety. As the enzyme is inactivated, the reaction which it catalyzes slow down and ultimately stop. Low temperatures generally do not destroy the enzymes. Most probably the low temperature it is not enzyme destroys. Its activity returns on heating to optimum temperature. Always the temperatures 60 to 80 degrees Celsius permanently destroy most of the enzyme. Some of the enzyme except fat hydrolyzing or inactivate at 0 degrees Celsius. It may be active at minus 15 degrees Celsius, and this fat containing food may deteriorate when stored below 0 degrees Celsius. The optimum temperature of an enzyme may be defined as the optimum temperature at which its activity is the maximum. The exact value of the optimum temperature depends upon the nature of the individual enzyme. For most of the body enzymes under ordinary conditions in near the body temperature. This graphical representation show that the optimum temperature. Some of the enzymes that may be needed for high temperature. It's applicable, that is between the temperature and increasing enzyme activity. The optimum it occurs in the 40 degrees Celsius. pH. The reaction velocity of an enzymatic reaction is very dependent upon the pH of the medium. Most probably all the molecules having some specific pH value. Based upon the pH, it determines the acid, base as well as the neutral. Normally the pH 4 to 9 values outside which the enzyme will not function. Optimum pH, the optimum pH at which the enzyme has maximum efficiency in catalyzing the reaction. Some of the example, the pepsin, the like a proteolytic enzyme, the trypsin, the pepsin having the 2.0 pH, trypsin 8.0 to 9.0, urease 6.8 to 7.0, salivary amylase 6.8. So most probably the enzymes, some of the enzymes they having the optimum pH like 8, the pH 8 having the optimum pH, the activity of the enzymes in increasing the activity of the enzymes. Substrate concentration is also one of the factors affecting enzyme activity. With a fixed amount of enzyme, the reaction rate is proportional to the substrate concentration. But this is the true to certain concentration of substrate do not further increase in the velocity of the reaction. There is no, once they are fixed in the particular place, there is no any decrease or increases. The reaction it occur in particular same places. So this graphical representation show that the substrate concentration as well as the velocity. Initially the enzyme and substrate is there present, there is no any reaction it's occur. They increase the velocity by the presence of the substrate concentration. One or two molecules they are binds, the complexes of enzyme substrate complexes will be formed. After the maximum velocity will obtained means they are the fixed ranges and the fixed temperature, fixed concentration will occur. There is no any changes, decreases as well as the increases. So these are the one of the type of substrate concentration in the factors affecting enzyme activity. Next one, biological functions of enzyme. Always the basic biological function is nothing but it is involved to the biocatalytic chemical reactions in the living things, especially for the body. Enzymes include detoxification, muscle building, and breaking down food particles during digestion. So the digestion process is also the enzymes, it's a main role, like a pepsin, trypsin, chymotrypsin, all the type of enzyme. It involved to the breakdown of the molecules. Enzymes actually accelerate the rate of a chemical reaction to support to the life. Enzymes very helpful in performing important functions of our body. Enzymes, it's a main role, it is a biocatalyst. It increases the speed of the chemical reaction by applying of the little amount of the energy to occur in the reaction process. And also the enzyme, it is involved to substrate specificity. So like in for example, the lactose means they needed for enzyme lactase enzyme based upon the substrate specificity they are needed for some specific type of an enzyme. It's only they break down and easily conversions of product with the help of the lactase enzyme in the substrate of lactose. And also this specificity ensure that the correct reactions occur in the right place at the right time. Yes. Lock and key model. So the next biological functions of enzyme. Lock and key model. Enzymes and substrate fit together like a lock and key mechanism. They already you know that lock it act as a enzyme, key it is act as a substrate. Once the key is fixed to lock, they easy to open. Only the correct shape and size of the key only open to the lock. The next one, some of the enzymes having the regulation process. So they need, especially the human cells or yeah, all type of cells, they needed for some specific regulation process. It's important. So these regulations only they involved to the metabolic pathways. It's a response to the organisms needed. And also the temperature sensitivity. They contains a high level of temperature. It leads to the destroy the some of the enzymes. So high temperatures can denature enzyme, which while low temperature can slow down their activity. So the optimum temperature will be maintained. They work is very efficiently. And also the pH, it's also important. They may be pH will be increases as well as it decreases. Always you know that optimal pH at which they function best. There is any changes it occur means there is enzymes also changes in the nature. Coenzymes and cofactors, already you know that the cofactors it is a conversions of inactive protein to protein. These small molecules helps the enzyme carry out their catalytic function. Enzymes also involved to the metabolic pathways, all the carbohydrate, protein, lipid, all the enzyme and controlled and regulated. Enzymes also involved to the inhibition process like a competitive, non-competitive, as well as the enzyme inhibition. Enzymes can be inhibited by specific molecule which can act as a competitive or non-competitive inhibitors. This inhibition is a mechanism to control enzyme activity. Enzymes also involved to the enzyme kinetics. Enzymes follow Michaelis-Menten equations, that is a Michaelis-Menten kinetics, which describe the relationship between the substrate concentration and the rate of the reaction. So this enzyme kinetics is also helpful for the some of the scientists and researchers to understand the enzyme behavior and optimize the reaction condition. Thank you all.